Welcome, everyone. My name is Agam Patel. I am the Associate Director here at UCR's Palm Desert Center, and it is my privilege to welcome all of you to the first of our four-part lecture series called Exploring Our Aging Brain. This is going to be an exciting lineup, and you won't want to miss it. But before we begin, I'd like to thank our Palm Desert partners who are instrumental, as always, in contributing towards programming like this. If you or someone you know would like to learn more about our partners program, we'll put a link in our chat momentarily, so look out for that. And I also want to thank our mighty Palm Desert, UCR Palm Desert team, without which none of this would be possible. So thank you. All right, let's get started, shall we? This program is brought to you in collaboration with UCR's Aging Initiative. And moderating tonight's discussion will be Dr. Aaron Seitz, Professor of Psychology at UC Riverside and Director of UC Riverside's Brain Game Center for Mental Fitness and Well-Being. Take it away, Aaron.
Great, thank you so much, and wonderful to have everybody here in the audience. And so as Agam mentioned, this is part of a 4-talk series on exploring our aging brain, which will be fourth Wednesday of each month. And I just want to tell you a little bit about the UCR Aging Initiative. And so this is a new group at UCR where we seek to create a world-class institute on healthy aging on the UCR campus, targeting to be a center of research, education, and a hub for the community. So our vision is to create a uniquely integrated program that considers aging from molecule to individual to society. And we seek to understand the integral role of human diversity in aging. This initiative currently involves over 60 faculty and staff and representations across all 7 campuses or 7 colleges of the UCR campus. So the focus of this first session, this series, is how does the aging brain learn, remember, and create? We're going to have 2 speakers. The first is Ilana J. Bennett. The second is Weiwei Zhang. I'll first introduce Dr. Bennett, who's going to give a talk on memory and the aging brain. And so Dr. Ilana Bennett is an assistant professor of psychology at the University of California, Riverside, and co-director of the UCR Aging Initiative. She received her bachelor's degree in cognitive science from University of California, Irvine, a PhD in lifespan cognitive neuroscience from Georgetown University, and completed postdoctoral research at the University of Texas at Dallas and University of California, Irvine. Her research has been continuously funded by the National Institute of Aging and seeks to advance our understanding of the way we acquire, retain, and retrieve information. She is particularly interested in how these learning and memory processes are affected in aging and studies their neural substrates using magnetic resonance imaging, often called MRI. And with that, take it away, Lana.
Thank you so much, Aaron. And thank you everyone for joining our talk series. It's very exciting to have you all here. Tonight, I'm going to be sharing, as Aaron mentioned, some of my work examining memory and the aging brain. So if you're anything like me, when you hear the words memory and aging together, you might think straight to something like dementia. And in fact, my own work was initially motivated by aiming for a better understanding of dementia. How could diseases like Alzheimer's disease rob us of our memories, and what can we do to stop it? But I'll acknowledge that more recently, my work has been motivated by understanding the healthy or normal aging brain and what memory is doing, how these normal changes in memory, let's try that again, how memory changes as we age normally. And this can really help us better identify when things become abnormal. Ooh, I'm not advancing. Here we go. So we're all familiar with this notion that our memory declines as we get older. And if I look within my own blood relatives, in general, my sister and I have better memory than our parents have better memory than their parents, and in turn, my sister and I have better memory than our grandparents. But it's important to also consider and also acknowledge that memory varies across individuals within the same age group. So my grandmother Lily on the left had better memory than my grandma Evelyn on the right. And in fact, my sister has significantly better memory than me. And she's 19 months older than me. So it's really not just about age. There's also these individual differences that we need to consider. So what I plan to show you tonight is how these individual and age-related differences, they don't just vary across individuals and age groups, they also vary for different forms of memory. And we think this has something to do with changes in the brain across individuals and across the lifespan. So I've broken the talk down into 3 different parts. I'll share some of our work in which we look at which forms of memory are spared and which forms are impaired as we get older. I'll then tell you a little bit about what happens to our brain as we age. And then I'll bring this together in the last section where we look at relationships between brain aging and memory performance to better understand if brain aging contributes to these declines that we see with age. So let's start with this first question: which forms of memory vary in age? And what I'm going to do is a little assessment of your memory. So if you want, you can answer in the chat, or you can just answer to yourself. I'll give you just about 3 seconds to come up with an answer for some of these questions. So the first question is, what does ruminate mean? So hopefully most of you immediately thought of the answer to think deeply about something. And those of you of a certain age might also have come up with the definition of chew the cud. And it turns out that younger adults are going to be much worse at this. First of all, they might not know what ruminate means at all. And they will certainly not come up with a definition of it being chewing the cud. And that's because things like semantic memories, these memories for facts and information, are preserved in aging. We find that older adults actually have better better vocabulary than younger adults. And they also have better general knowledge about the world. And this is really consistent with the anecdotal saying that we get wiser as we get older. These semantic memories are actually preserved. But let's try another example. Okay, what I'm going to do here is read you a list of words, and I want you to remember them. Bed, night, tired, blanket, dark, moon, nap, dream. Okay, there's lots of ways that I could probe your memory for this list of items. And all of these different ways that I could test your memory for them are going to tell me about your episodic memory. So this is how well do you remember things that happen in a particular time or a particular place. You've heard all of these words before. So the goal is to really remember, do you remember them from this instance, from this episode? Do you remember hearing them just now? So if I were to ask you, was the word dream on the list? Hopefully, most of you could remember that it was in fact on the list. And that's because recognition of these episodic memories is relatively preserved in aging. I'm asking you about a specific word like dream, and that can serve as a clue to help you solve this question, especially if the word dream sounded a little bit familiar. But what happens if I ask you which words were on the list? This type of free recall should feel much harder than the recognition from the previous slide, because now you need to recollect exactly which words you heard from this specific list. There's no longer any clues to help you out. And what we find is that this kind of recollection of your episodic memories is significantly impaired in aging. This recollection requires really detailed, precise memories, in this case, of the words that were on the list. And that precision of your memory is something that degrades with age. So you hear a little bit more about this from Dr. Zhang in the next talk. Okay, this is the last question I'll ask, I promise. Was the word sleep on the list? Okay, this was a tricky one, I'll acknowledge, because what I did was I gave you a lot of words that are really similar to the word sleep, even though sleep was not on the list. So I said bed and night and nap and dream, but I never actually said the word sleep. So apologies for being a bit mean here, but it's a really great way for me to demonstrate that discriminating between very similar episodic memories is significantly impaired in aging. So just like the recollection that I showed you in the previous slide, What discrimination requires is, again, that you have this very detailed, very precise memory so that you know exactly which words you previously saw in this exact same context. And it's these really detailed memories that become less precise with age. We end up shifting to a more gist-based memory, like the gist of the words that you heard had something to do with sleep. Okay, so this is what the data looks like, summarizing the effects that I just talked about. These are data that we collected looking at the three different forms of episodic memory. So the vertical axis on each of these graphs is showing you performance on the recognition, recall, or discrimination tasks. And on the horizontal axis, I'm showing chronological age. In this study, we looked at 120 individuals who were aged 20 to 89 years old. And so what we can see is on the left, for example, recognition performance is relatively preserved. So both individuals in their 20s, 50s, and their 90s were able to say whether dream was on the list. If we look in the middle graph here for recall performance, this is if I were to just ask which words were on the list. Now we see that this begins to decline with age. That regression line has a bit of a negative slope to it, and that indicates that there's a significant negative effect of age here. And that slope is even greater for the discrimination. So older adults are much worse at telling me whether sleep was on the list, discriminating whether sleep was one of those words on the list. But one of the things that I also want to point out here is that although recall and discrimination memory were impaired as we get older, you'll notice that there's differences in when that impairment begins. And so for recall, it really doesn't start until we're about age 50 and over. For discrimination, it actually starts in our 20s. Okay, so this again goes to this idea that it's not just about age. There's more context that's needed, in this case, the specific forms of memory that are impaired. Okay, so in this first section, I showed that some forms of memory are spared in aging, like recognition, and other forms are impaired in aging, like recall and discrimination. And now I'll switch to the next section where I talk about what happens to our brain as we age. I do just want to say that there's lots of things that I could look at here, lots of mechanisms that we think might explain why some forms of memory vary with age and others don't. So we could look at lifestyle factors like sleeping or alcohol consumption or diet, or environmental factors like pollution or social interactions. But as a cognitive neuroscientist, I'm really interested in understanding how the brain structure and brain function changes with age and contributes, in this case, to differences in our memory. So we study brain aging using magnetic resonance imaging, or MRI. And what we do is invite participants to come into the scanner, and they lay down on the bed in the middle here, and they're in there for about 30 to 60 minutes. Sometimes we ask them to do a task, so they might push buttons, and other times they just lay around relaxing. And we can acquire a bunch of different scans in any given scan session, and each of them can tell us something different about the brain. So I have various studies going on right now in which we're looking at the effects of age on things like tissue microstructure and macrostructure, on accumulation of iron, myelin content, perfusion of blood, neuroinflammation, as well as neural activity. For this talk tonight, I'm going to focus on the first two, tissue macrostructure and microstructure. So tissue macrostructure is essentially the stuff that we can see. So what you're looking at here is an MRI scan from a single individual, and I'm showing you just a single axial slice as though we're just cut open their brain about here. The front of their brain is pointed to the top of the slide, and the back of their brain is pointed to the bottom of the slide. And so it should be really obvious that there's two different tissue types. There's a much whiter looking tissue, and that's referred to as white matter, and then a darker tissue referred to as gray matter. And these correspond to different parts of neurons that do different things. So the white matter is comprised of axons that connect different parts of our brain. And the gray matter is comprised of their cell bodies and dendrites that actually process information. So if I was to compare tissue macrostructure in this 69-year-old female relative to this 29-year-old male, there's a couple of things that are really obvious even to the naked eye. So the older female has what we call enlarged ventricles. Those are the fluid-filled spaces in the brain. She also has sulcal widening, and this is an expansion of the valleys that you can imagine in between all of the different grooves that cover our cortex. And so both enlarged ventricles and sulcal widening are indicators of atrophy or a loss of tissue as we get older. But it's not just about these macrostructural changes. And it turns out that there's lots of things that can happen to our brain at the cellular and molecular level. These are things that would be happening within a voxel, which is the imaging equivalent of a pixel in an image. And so the way that we can assess tissue microstructure is using diffusion MRI. And what we do here is we're looking at the jitter or diffusion of water molecules. And the idea is that these molecular water molecules, they're going to be diffusing differently depending on whether and how they're obstructed by the presence of tissue. So you could imagine that if there is a loss of tissue, a loss of neurons or neural structures, that water is going to diffuse more freely in all directions. We can also look at different diffusion metrics. And if, for example, we see an increase in diffusion within an intracellular compartment, that might indicate that we have inflammation, such as swelling in our glial cells. So for this talk, I'm not going to focus on the specific diffusion metrics, although you will see them in the slides that I present. Instead, I will talk more generally about diffusion and whether it's good or bad, whether it's better or worse. So here I'm showing data from one of our studies in which we had adults across the lifespan undergo diffusion-weighted imaging. And now I'm showing you 3 slices of the brain from the bottom to the top as you go from left to right. And all these little voxels or pixels that you see in blue here indicate regions of the brain, regions of this white matter, where diffusion got worse as we got older. So there was more water diffusion relative to a younger adult. And the scatter plot on the right is showing the exact same data. So you can see age on the horizontal axis and a measure of diffusion, in this case, fractional anisotropy on the vertical axis. And one of the other things that I want you to take from this slide is that these blue voxels are everywhere, right? We really see these effects of age across all white matter in the brain. But one of the questions that we had is whether some regions are more vulnerable to aging than others. And so my graduate student, Jenna Merenstein, followed up our original study by looking at changes in tissue microstructure within older adults. So here she's just looking at individuals who are 65 to over 100 years old. And what she found is that these negative effects of age on white matter are the largest for tracts emanating from the hippocampus. So that middle image shows the blue dots, which is part of the tracts that she was looking at. And on the left, I've shown the hippocampus in this sort of cartoon image highlighted in yellow. The hippocampus, we think, is a really important structure given its role in memory. It's also one of the structures that's affected in Alzheimer's disease. So we weren't terribly surprised to find that integrity of tracts that connect the hippocampus to other parts of our brain is particularly vulnerable to aging. I then had an undergraduate honors student also follow up this work, looking at the effects of age within gray matter of the hippocampus. And so you can see the hippocampus shown here in 2 different slices of the brain in light blue. And what she found is that there were age-related increases in diffusion across multiple measures of, from our diffusion MRI scan. And in fact, it's the only brain region that shows increases across all of these metrics. And we think that this is an indication of the hippocampus of neuroinflammation within the hippocampus, and the hippocampus being particularly vulnerable to that neuroinflammation. And so later in this talk series, you'll actually hear more from Dr. Monica Carson and Chandra Reynolds about the role of neuroinflammation in aging. Okay, so I've already told you that some forms of memory decline with age compared to others, and that aging is accompanied by declines in both tissue white matter and gray matter microstructure, and that it's particularly vulnerable in these hippocampal regions. So how can we put all of this together to better understand whether brain aging can explain these declines in memory? And we do that by looking at relationships between measures of tissue structure. In this case, on the horizontal axis, I'm showing integrity of one of those hippocampal white matter tracts, the fornix. And I'm showing you the relationship to performance across those three different memory tasks that I mentioned in the first section of the talk. So what we find for recognition is that there's no significant relationship between microstructure of these hippocampal tracts and performance on that task. But for both recognition and discrimination, we find significant relationships such that better diffusion is related to better performance on these tasks. And it turns out that these effects are independent of age, meaning that having better white matter microstructure with better connections between the hippocampus and other parts of our brain is going to relate to better memory performance regardless of how old you are. So this is really one of those individual differences. This might explain why my sister has better memory than me if the integrity of her fornix is better than mine. This finding also supports the idea that these memory processes, at least for recall and discrimination, rely on the hippocampus, reinforcing the idea that this is an important structure for memory. My other graduate student, Anu Venkatesh, also followed up the work in discrimination by separately examining how gray matter microstructure within the hippocampus relates to discrimination performance. And what she found is that better performance, better discrimination memory, was related to better diffusion, in this case, an intracellular metric, diffusion metric within the hippocampus. But this relationship was only significant for younger adults. It wasn't significant for older adults. And we think that this age moderation might actually explain some of the age group differences that we see in memory performance. So you can imagine that as we age, our hippocampus microstructure degrades. And as a result, we might not rely on this brain region as much in order to perform this same task as younger adults do. So let's put this all together. What I hope I've shown you tonight is that some, but not all forms of memory are affected by aging. So recognition is relatively spared, whereas recall and discrimination performance are impaired. I then showed you that aging is accompanied by declines in tissue microstructure throughout the brain, and that these effects are especially large for the hippocampus. And finally, I showed that hippocampal microstructure contributes to both individual and age-related differences in memory performance. And this was specific to recall and discrimination, not necessarily for recognition memory. And again, this brings us closer to better understanding normal brain aging and its contribution to memory declines in a normal, healthy individual. And this can lead us to better detecting when there's a problem, better diagnosing individuals who might be at risk for, if not having dementia. And these imaging and behavioral metrics can also serve as targets for interventions and therapies. So with that, I would like to thank especially the participants who made this work possible. And I bolded my students and collaborators who were involved in the projects that I mentioned, as well as our funding from the NIA. And I would also just invite you to reach out to us if you have any questions about what you've heard here, or if you would like to get involved in any of our work. Thank you.
Great. Thanks, Dr. Bennett, for the wonderful talk. For those of you in the audience, So we're going to be holding questions to the end, but so that you don't have to remember your questions, given that we're doing a series on memory, you can post them now to the Q&A or at any point that you have a question, and then I'll read them off when we get to the end so that you could get the answers you're waiting for. But without further ado, I want to introduce the next speaker, Dr. Weiwei Zhang. He's going to give a talk on a neurocognitive model for foggy memory and aging. And Dr. Zhang received a bachelor's degree in chemistry from Nankai University in China, then a master's degree in cognitive neuroscience from Beijing University, then a PhD in experimental psychology from University of Iowa. And then after completing an NRSA postdoctoral fellow at UC Davis, he joined the faculty of psychology at University of California, Riverside in 2012. The research program in Dr. Zhang's laboratory has been funded by the National Institute of Mental Health and the National Institute of Aging, and it focuses on perception, attention, memory, and higher order cognition in healthy and clinical populations. And Weiwei Zhang, please take it away.
Thanks so much, Dr. Seitz and Dr. Bennett for that great talk. Also like to thank our partner at the UCR Palm Desert Center, and thank you all for coming and joining us today on Zoom. I'm going to talk about foggy memory with age. So here I'm playing a short video clip. And also with this video clip, I want to demonstrate why we care about memory. So we care about memory because memory are at the center of the higher cognition. For example, here this goalkeeper clearly has the knowledge about what to do in this kind of situation. So that's what we call book smart. So the knowledge is drawn from what we call long-term memory. In contrast, the implementation or the presentation of this knowledge seems to be lacking here. So the goalkeeper is doing something that's inappropriate. So he jumped even after the ball bounced off his face. So these two types of intelligence are related to two types of memory systems. So the book smart seems to be closely related to what we call long-term memory, and the street smart, which is lacking here, seems to be related to what we call short-term memory. And what are these two memory systems? So here, the cartoon on the left side demonstrate a memory system that we use to maintain information in mind for a few seconds or minutes for the current moment. So here, this individual was asking, was making a statement, my memory is so bad, and a few seconds later during the conversation, this individual pretty much forgot about what's bad. So that's what we call short-term memory. And in contrast, we also, so everybody seems to have this, this ability to maintain information over the entire lifetime. We are still able to remember the childhood stories and friends. And in contrast to short-term memory, the long-term memory seems to have the ability to represent, to maintain and store a vast amount of information over the lifetime. And in sharp contrast, short-term memory is highly limited by its storage capacity. Decades of research in psychology and neuroscience demonstrate that we can only maintain a small amount of information in short-term memory over a few seconds. To make this more clear or concrete, I'm going to show a few demonstration trials. Let's see how this works. So I'm going to present two sequential displays of colors. I apologize if you are colorblind. And your task is to memorize the colors, all the colors, if possible, from the first set. Then after a few seconds, you're going to see a second display of colors. Your task is to report whether you see a change color in the second display relative to the first display. All right, so we're going to try this 3 times. Let's start with the first attempt. All right, ready, set, go. These are the memory colors. Try your best to memorize these 2 colors and then hold your memories for the next few seconds. Then here comes the test display. Your task here is to report whether you see a changed color in the second set of colors as compared to the first set of colors. Feel free to type your answer in the chat. So if we can go back in time, so here is the first set. So this color red on the bottom left changed to color yellow. So I'm pretty sure the majority of us can remember 2 colors. All right, so that's the first attempt. Let's do one more time, the second attempt. Ready, set, go. These are the memory colors. Try your best to memorize them all if possible. Then hold your memory again over the next few seconds. Then the test. So do you see a color, change color in the second display relative to the first display? The research in psychology in the past 20 and 40 years demonstrated that most of us are able to remember 3 to 4 colors in short-term memory. So this should be doable. Let's try the last time. Ready, set, go. Memorize all these colors if you can, as much as you can. Then hold your memories over the next few seconds. All right, then test. And don't panic if you cannot detect the changed color in a second display. If we can go back in time, so here the color like orange at the top changed to color pink in a second display. So the majority of us cannot do this trial because the amount of information we need to maintain over a few seconds in short-term memory is too much. It's not our short-term memory system can handle. So yes, unfortunately, we are able to only maintain a small amount of information in short-term memory. And then in the cognitive aging research, we had this strong focus on short-term memory capacity, on how much information we can maintain over a few seconds in short-term memory. Here is a typical finding. Apologize. All right, so here is a typical finding in the literature. This finding has been replicated many times, including some publication from my lab as well. So here on the x-axis is the age. On the y-axis is the short-term memory task performance. What's typical in this kind of situation that the observation is that our short-term memory task performance declines with age, especially the amount of information we can maintain over a few seconds in short-term memory declines with age. So even though, as I mentioned earlier, this finding has been replicated many times, but unfortunately, this is not consistent with phenomenal experience to some of us. So when I was in college, when I was taking all the classes required for my degree, my memory is super clear. But these days, this would be what my memory looks like. They are blurry, they're foggy. Here are some artistic representation of our foggy memory. So if memory over age is becoming more and more blurred, what's the problem? So why should we worry about it? So for example, if this is what my memory looks like for a student in my class from the fall quarter, then with this blurry memory, I cannot tell whether this student is Sarah or Lisa, because they are both of them are likely to produce this kind of blurry memory. All right, but before we conduct research to understand why memory is becoming foggy over age, first thing we need to do is to measure the foggy memory. So we develop a task, a simple, very simple task to do so. I'm going to show a simple trial sequence. This is basically exactly what our participants will be doing. We start with a fixation. Oops. Then we present 3 colors on the screen, just like the demonstration I did earlier. So here, the participant's task is to remember the 3 colors as precisely as possible. Then they hold their memories over the next few seconds. And then comes the test. So here we probe, see whether I can turn on the cursor. Apparently not. All right, so here we probe one of the 3 colors. And I reviewed the answer. Apologize, I'm going back in time. All right, so here we probe one of the 3 previously shown color. And the participant task is to pick a color on the color wheel's continuous color wheel to match their memory for this color. Here, let's see if I click here, and that's what our memory for this color is on that trial. Now we also show to the participants the actually presented color by the arrow here. So the difference in this case, the difference between the color the participant chose on the color wheel and the actually presented color represent how precise our memory is. So here we have 2 scenarios or from two participants. Let's see, the first participant over many trials, over many attempts, produce a chose color. This color is represented by the white dots. These dots are closely clustered around the actually presented color. And in contrast, a second participant over many trials pick the colors that are spaced out on the color wheel. So this is kind of like the dart game, right? So we want good clustering on the left side over the spaced out clustering of the darts on the target on the right side. So in the first scenario, the performance on the left side represent more precise memory, and the performance on the right side indicates less precise memory. So that's exactly what we did in this study published in Journal of International Neuropsychology in 2019 in collaboration with our collaborators at Michigan. So here on the x-axis is the age, on the y-axis is the measure that we actually had from this specific task. So the y-axis is actually represent imprecise memory. So the higher this number is, the crappier memory is. And each individual dots represent the performance from individual subjects. So for example, on the bottom left side, these folks, younger folks, seem to have higher memory quality. In contrast, these folks on the right side, top right, seem to have worse or crappier memory. And we did some statistic tests, and what we found is that the age is a good predictor of the memory imprecision. So the older we are, the more imprecise our memory is. What this finding suggests is that over age, as we are getting older, our memory gradually fades away. All right, so why do we care? So why do we care whether, with age we gradually lose memory quantity or memory quality? We have several reasons to do so. First, memory quality reduction with age may serve as a predictor of dementia. It can also inform us how brain ages. So, with the understanding of the neural mechanism and cognitive mechanism of memory reduction, especially in memory quality, it can give us some mechanism and ideas for intervention. I think I'm good in time. Lastly, we appreciate the funding support. The work is done by the team. So here are the photo of the team, including the research staff, graduate students, and postdoc scholars in the lab. And thank you all.
Great, thanks, Dr. Sheng, for a wonderful talk. So while people in the audience consider questions that they could post, I'm going to actually start off just with one question that I have that's kind of burning. And that's, so Dr. Bennett kind of talked about kind of discrimination memory. And Dr. Zhang, you talked about foggy memory. I'm just kind of curious if I have trouble remembering kind of one of these exact colors. And so, you know, my memory is foggy for the color memory. Does that mean that I'm going to get confused and say that sleep was in Dr. Bennett's word list?
I would think so. So one of the consequence of the imprecise memory is that it may introduce a false memory. So the experiment paradigm and finding Dr. Bennett presented earlier seem to indicate with age, the false memory is likely to grow. And I do believe the false memory and the likelihood to have false memory is linked to the qualitative aspect of memory.
I agree.
Great, thanks. So let me take some of the ones from the audience. So the first question is from Gail Heitman. And so what effect does studying a new discipline, e.g. new language, philosophy, have on the aging brain for the elderly?
I can take a stab at that first. It's good for you. It's really good for you. So we find that anything that is a challenge is going to improve brain function, and maybe not just for the thing that you are getting trained on. So if you're doing something that you've always done, you're doing a crossword puzzle and you're already exceptional at crossword puzzles, it's not a challenge for your brain, and you're not going to really see any improvements or any transfer to other skills that you might be interested in preserving or improving. But if you do things like learning a new language or studying a new topic, that's really the best thing that you can do. Maybe the only thing better than that would be exercise. So I think, yeah, we certainly see improvements in both neural measures and performance measures when you take on a new challenge.
So a similar question. So clearly there's an interest in memory improvement in this audience. Is from Colleen Crowley. Does Prevagen or other supplements help memory?
Wait, I'm also happy to take it, but you could, do you want to jump in or?
Go ahead.
Okay. So the literature on supplements is very unclear. Things like Prevagen, I would look very carefully on the internet for any evidence that they're claiming about improvements related to those supplements. There are certainly certain things that we can do. For example, antioxidants, which are present in blueberries, have been shown in animal studies to show improvements. But the amount of blueberries that you would have to eat to show any benefit, it would just make you sick. So it's really not, I would just be very wary. There's not anything that the medical establishment would agree on or the scientific field would agree on as a supplement that has scientific proof of its benefits over other things. I think the only thing that people would agree on, as I mentioned, is exercise.
Okay, next one Is the aging population that was studied active older adults or retired? Or does it make a difference? Can you just tell us something about, I guess, individual differences that might be important with these results?
Wei, you go ahead.
Yeah, we have some unpolished results from the lab. Yes, the lifestyle does matter. So individual differences in memory performance across short-term memory, long-term memory can be predicted by differences in lifestyle and many other factors as well.
Yeah, I'll just, oh, sorry. Go ahead.
Sorry. Yeah.
I was just going to add to that and say that for most of our studies, we have included older adults. Most of them are retired. Some of them are still working. But I think something that your question is getting at is really interesting in that there is somewhat of a selection bias. So the people who are looking for opportunities to engage in research are different from individuals who might be housebound or have significant enough cognitive impairments that they wouldn't be able to schedule a testing session. And so it does tell, you know, the kinds of studies that we've been doing in some ways are a little biased to a more active, engaged individual. Although there are other studies that do more community-based sampling, population-based sampling that wouldn't necessarily have that bias.
And I could just add that kind of one of the goals of the Aging Initiative is to do more communities-based sampling in our region, including Palm Desert. Next question is, given the way the brain ages, if we continue learning throughout life and increase learning as we age, are we likely to do as well with the learning later in life? Or is it more likely that we'll struggle with learning as we age?
I'll take this one. Hi, Stephanie. I happen to know Stephanie. What I think your question is also getting at here is this idea of individual differences. So if you are someone who engages in learning throughout your life, chances are that you will continue to do well with learning throughout your life. So we find that if you are a high functioning young adult, you will continue to be a high functioning older adult. And the same if you are a low functioning, if you are not learning well as a younger adult, you are going to be more likely to struggle as you get older. So I think in continuing to engage in learning throughout your life, and as I mentioned, continuing to challenge yourself. So don't just go over the same types of things that you already know, are the best things that you can do to maintain cognitive health.
Right, and one thing I want to add, so I saw Dr. Rachel Wu earlier in the participants. So in this talk series, I don't remember the third session or the last session, Dr. Wu will be talking about learning as intervention.
Great, nice plug. Okay, next question. I am finding in my mid-60s, they have constant flashbacks to the past, including childhood, that are vivid and accurate. What does this say about my memory? Is this part of the process of losing it?
That's a great question. So the memory, the subjectively felt vividness of memory, recollection, remembrance seem to be supported by the qualitative aspect of memory that I talked about earlier. Sorry, I didn't get the second part of the question. Is this part of the processing of losing it?
Yeah, so if you're starting to remember your early life memories, does that mean that you're losing your ability to remember new things?
That's a great question.
How I took the question.
Dr. Bennett?
I actually do not, I'm racking my brain here. I don't think so. I think that the, I don't know if I'm going to, perseverating is not the right word, but sort of reminiscing about highly salient memories is a different process than not being able to learn and remember new things. So I think these are two different things that would be going on.
But I think, the flashback to vivid and accurate childhood memories may be an indicator of a healthy brain. So it does at least suggest the memory structure Dr. Bennett talked about earlier in the hippocampus structure in the medial temporal lobe seems to be functioning very well. So that's, to me, in my opinion, that's good.
Very interesting question. So next one is by Kevin Kigma. If an aging person only has sufficient memory for daily living activities, does memory loss present other problems? Does memory loss usually lead to anxiety or mood problems?
I actually think that the anxiety and mood problems are separate, except that when you do start to lose your memory I'm thinking in the course of Alzheimer's disease, not understanding how things have changed can create some anxiety. So for example, you could imagine that at night you have dinner and you have a glass of wine and you don't put those dishes away that night. In the morning you wake up and you see glasses of wine out and dishes and you're confused and you think that someone might have broken into your house or who is up eating in the middle of the night. And so there's, in that way, when you have dementia, the memory loss can create anxiety. But I think that there's other mood problems that also accompany the progression of dementia. If you're talking about just normal memory loss, I think we have just a healthy amount of anxiety about any form of memory loss being a sign that we are going to progress on to dementia. And I think that older adults have this fear more than younger adults, even though we experience plenty of memory lapses in our youth, and we just attribute it to stress or drinking or other things. As we get a bit older, we attribute it to, this must be an early sign of dementia.
Yeah, one thing which I might add actually is of a colleague, Nicholas Turk-Brown at Yale, who's done some studies in people who have bilateral hippocampal damage. And so they have very severe interrogated memory loss. So they can't remember new things. But they're also some of the happiest people that he's ever met. And, there are definitely trouble points in terms of there are moments where they realize that, for instance, one patient, on a daily basis would be told that her father was dead and, would grieve and then forget that and be happy again. But there are some cases where, extreme memory loss can actually promote extreme happiness. And actually, a question I actually wanted to ask Wei Zhang is I have a foggy memory of you having a study where you found that at the same time that memory is becoming more foggy, that creativity wasn't being improved. I'm just wondering if you could speak to that, because I think there are some positives that accompany, some types of memory loss.
Yeah, that's a good point. But if I may, I would rather not talk about a specific study and the findings. I do want to follow up on this. So yes, so in some of our studies, published studies, we did find that with age, we have the increases in memory loss and also increases in anxiety, depressed mood. So this seems to go side by side. But the question here is that is there any causal relationship? So does one functional decline leads to the other? So that's something that we don't really know. So these two problem, maybe a memory problem and the mood problem, may be related to the decreases in memory, in sleep quality with age. So there are a lot of possibilities here.
Thank you. So next question is by Jacqueline Alba. So as you mentioned, exercise would help improve. Does diet also implement change? What findings do you know
of? So my understanding of I would put diet and supplements in the same bin. So you will find one-off studies showing benefits of a Mediterranean diet, for example, or an Atkins diet, or certain foods that should be eaten and other ones that should be avoided. But most of these do not hold up to rigorous scientific evaluation, and there's really no consistent findings across studies. So for every study that says, a glass of red wine will benefit you, there's another one that shows that it doesn't have any effect, or that it would have a negative effect. And so I at this point just don't think that there's enough work showing support for any dietary, any specific diet that you would choose.
I think maybe the one exception I might point out is extremely poor diets are harmful.
Yes, malnutrition should be avoided.
Okay, next one from Patricia Liu. So what about online brain game apps such as Luminosity?
This is perfect for Dr. Seitz to respond to.
So yeah, since I run the UCR Brain Game Center for Mental Fitness and Wellbeing, we actually do a lot of research on these types of apps, and that the research is very mixed. So that with some of the companies, Lumosity is one of them. They've been criticized that the evidence is not clear to the extent that they're actually beneficial to the extent that they claim. At the same time, if you start looking at individual studies, there is some pretty exciting evidence that, at least in certain people, certain types of brain training apps can have positive effects. And the place where it's really frustrating, especially to someone like myself who does research in this area, is that, well, I think there's a lot of promise of these types of apps. The research isn't sufficient at this point to have a good sense of which people are going to get what benefits from what of these apps. And so, my general advice is that, similar to lots of, diets or supplements or other things like that, you could try them out cautiously, and there is some possibility that you might get benefits. But you shouldn't expect that, it's necessarily going to help you. And that also, if it helps someone else, that benefit might be specific to some aspect of that other person and might not apply to you. And so this is the big question for the research, is how can we do better science to be able to predict which types of apps will give rise to the benefits that different people want. And I think with time we'll do better, but right now, as I said, like any other type of self-help, you have to try things out cautiously.
So Doctor, on that note, Dr. Seitz, is it possible for you to post a link to your brain training games in chat?
They beat you to it.
Yeah, there it is.
Perfect.
And actually, one of the things that, we're actually recruiting for quite a few different studies. And so we have some studies that are focused more on vision training and hearing training, others on working memory. And all of our studies are really trying to get the type of evidence that we need to understand, which approaches might work better than others.
And I will just say you'll also hear more from Dr. Seitz later on in this series. So he will be presenting some of his work on vision and hearing. Mostly hearing, actually.
Yeah. Okay. So from Pam Muttner, what are the other possible complications of an increasingly impaired hippocampus?
That's a great question. I can take a stab at it to start. So as I mentioned, the hippocampus is thought to primarily play a role in memory. And one of the things that also fascinates me about memory is this idea that everything except for the immediate moment that we are in exists in our memory. So it really has all of our interactions, except for the immediate interaction that we are having with our world, in some ways relies on our memory for what we have done in the past. And so while it might just affect, we might think like, okay, I'm going to forget what my grandchildren's names are or what food I need at the grocery store. It can actually lead to some pretty basic functions that we're not able to do. So some of those movies like Memento, and there's a couple of other ones where you can see people who have complete hippocampal loss really just aren't able to remember what they're supposed to do when they wake up, what clothes they're supposed to put on, where to find those clothes, what house they're in. And so at the very extreme, it has an impact on every aspect of your life. For most people, it's the memory loss that you're going to experience is just a kind of a nuisance, right? And you'll just have to find ways to write little notes to yourself so that you remember certain things and look stuff up more than you might want to. Yeah, I don't know. Wei, do you have anything to add to that?
If I may, I would just like to highlight that the hippocampus, by the way, this is a great question. The hippocampus seems to be involved in many cognitive functions, including spatial navigation. So one consequence of hippocampal lesion would be the reduced ability with spatial navigation and spatial memory as well.
Great, thank you. So we have another question from Stephanie Hackney. So I've heard that we tend to recall things with strong emotional context. Is this supported by research? And if so, does this change as we age?
If I may, yeah, we do have some research showing that emotion can promote memory under some conditions, in some scenarios. But at the same time, strong emotional charged materials and context can also hurt memories. So there is a huge literature on flashbulb memory. So the idea is that the emotional experiences works like the flashbulb on a camera. It lights up everything in the environment so that you have better memory encoding of the information from surrounding. So that being said, yes, so strong emotional charged information can also hurt memory sometimes. And Dr. Bennett, you want to talk about aging? The second part of the question.
Yeah, so I was just thinking of some of the work by Mara Mather, for example, which shows that as we get older, that there really is a shift towards more positively, emotionally valent content. So if anything, I think that there's some evidence that the changes that you're going to see in remembering emotional things are going to be driven more towards positive emotional experiences.
Thank you. So last call for questions. I definitely want to thank the audience for a very stimulating set of questions. And then also the speakers for some wonderful answers.
Thanks, everyone. Thanks for joining us.
Thank you all.
And for those of you still here, this has been recorded, so it will soon be posted so that if you want to rewatch any part or share with friends, it will be available. And thank you for a wonderful night.